Let's start with a practice question. A 5-year-old girl is seen at the pediatric emergency department for non-communicating hydrocephalus. Which of the following is the most likely cause? And the choices are here, which you can read. To answer this question, there's a lot of things that we need to know and many terms that we need to understand. So the first thing that we need to know is, what is hydrocephalus? We'll talk about that in this lecture. And what's the significance of non-communicating versus communicating? So we'll cover that. And then in these answer choices, overproduction of CSF by the choroid plexus. So what's CSF and what's the choroid plexus? And how is CSF supposed to flow if this answer choice says it's obstructed? So we need to know what the proper flow is, and then what these things called arachnoid granulations are. For this choice, answer C, stenosis of the cerebral aqueduct. Well, what's the cerebral aqueduct? And then answer D, inflammation of the meninges. Well, what are the meninges? So we're going to answer all of these questions, and by the end of this lecture, you'll be able to get the correct answer to this practice question. So let's start with the meninges, this question number 8. So meninges are protective coverings of the brain, and there are multiple layers of these protective coverings. Their function is to cover and protect. You can see this drawing here of a dissection of the brain where the dissector is using a blade to cut through this outermost protective covering, which is actually quite tough and quite hard, and thus gets the name dura mater. So mater is mother and dura is tough. So this is the tough mother. Deep to the dura mater is the arachnoid mater. You can't really see it here, but it's covering the brain, and it's called spidery mother because it has a sort of web-like appearance. And then the most intimate, pia means intimate or gentle, the gentle mother is like shrink wrap, making the closest contact with the brain. As a clinical correlate, meningitis, itis means inflammation, so meningitis is inflammation of the meninges. It can be bacterial or viral or even fungal. Going off of that, you can have inflammation of the meninges, which is meningitis. You can also have encephalitis. So remember, encephalon is brain. So this is inflammation of the brain tissue itself. But you can also get meningioencephalitis, which is inflammation of both the brain and the meninges together. So let's look closer at these layers. This is a cartoon if you want to pause this and try to answer this on your own as active recall. This is a coronal section of the brain here, and we're labeling the different layers of the meninges. So number 1, in orange, which makes closest contact with the brain, you can see that this layer goes into the sulci and wraps around the gyri. That is the pia mater. And I show this illustration of the bed because I think these 3 layers are analogous to the 3 layers on the bed. The pia mater is like the bottom sheet of the bed. It makes close contact and wraps all the way around the mattress. The top sheet is number 2, that's the arachnoid, and it has this sort of spidery-like structure. These are called the arachnoid trabeculae, which kind of hold the arachnoid up and create a space here underneath or sub to the arachnoid called the subarachnoid space. So that's 2A. So this is the bottom sheet, the pia, the top sheet arachnoid, and you can see that the arachnoid does not wrap around the mattress. It goes over. Okay, and then the top layer, outermost layer, is the dura. That would be like the comforter. And the dura is made up of 2 layers. There's a periosteal closest to the skull and a meningeal, which is closest to the meninges. And those 2 layers can split, and they split in certain places. So they split in this what forms the superior sagittal sinus, and that ends up becoming a place where the venous blood and the CSF is drained. We'll talk about that later in the lecture. Are there any other potential spaces? Well, yes. So the subarachnoid space is the only real space that is present in a healthy brain. Now, when there is damage or bleeding, there can be 2 additional spaces that form, and that's why they're called potential. These shouldn't be there, but they have the potential to be there in the case of a hemorrhage. So here you can see that this is a space that's superficial to the dura, and that is the epidural space that is formed due to an epidural hemorrhage. This occurs in the case of a meningeal artery that ruptures, most likely due to trauma, such as the middle meningeal artery. So this is an epidural hemorrhage. Underneath the dura, deep to the dura, there is this subdural hemorrhage illustrated here. The subarachnoid space, remember, this is not a potential space, this is a real space, but it can also fill up with blood in the case of a cerebral artery or vein that ruptures, and there's blood in the subarachnoid space. We have another lecture devoted to hemorrhages and how these appear on CT scan that you can check out. Now, in the subarachnoid space, so this sort of light blue space, there are 3 things that travel. So you can see there are cerebral arteries, and cerebral veins, cerebral arteries and cerebral veins, but the other thing that's found in this space is cerebral spinal fluid, or CSF. So what is CSF? CSF stands for cerebral spinal fluid. It contains water, ions, and a small amount of plasma proteins with no or very few white or red blood cells. And so this is an ultrafiltrate of blood plasma. It's a clear liquid, and it has many functions. One of the most important is that it cushions the CNS. So the entire brain and spinal cord is surrounded in the subarachnoid space by this CSF that provides a cushion. Think about an egg floating in water. It's going to have cushion and protect the brain from like banging it against the inside of the skull. Also, it gives buoyancy to the brain, just like your effective weight is reduced when you jump into a swimming pool because of buoyancy. And this is important because there are many delicate cranial nerves that come off the bottom of the brain and are traveling through the skull in these tiny little holes. And you wouldn't want every time you go for a jog for the brain to be smashing on the bottom of the skull and crushing those delicate cranial nerves. So the brain floats in water inside the skull. It has cushion and buoyancy from the cerebral spinal fluid. And one of, I think, the neatest functions is this lavaging or rinsing of the central nervous system. This was recently discovered as a role of the CSF in cleansing the brain, similar to the lymphatic system in the body. And so and it involves the role of glial cells. So it was nicknamed the glymphatic system. So as you go throughout your day, the CSF that is around in the ventricles here diffuse and wash through the brain parenchyma and cleanse the interstitial spaces of the brain. It washes away byproducts of neuronal metabolism. When you sleep, it actually was just discovered recently that when you sleep, the interstitial spaces of your brain expand and you get increased CSF flow that washes out, again, those byproducts of neuronal metabolism. This is one of the reasons your head feels so cloudy and heavy when you pull an all-nighter, is that you're not getting that CSF lavage function that you would normally get when you sleep. So this lavage function happens all around the clock, but it's increased like threefold when you sleep. So CSF is made by the choroid plexus. This is a highly vascular structure that forms the roof of all the ventricles. We'll talk about the ventricles in just a minute in more detail, but you can see here in blue is the arachnoid. See how it's not going in deep into the crevices. In red is the pia, which is intimate associated. And then lining the ventricles are ependymal cells, which are glial cells. And when the pia comes in contact with the ependymal cells and with capillaries, it forms this choroid plexus structure that acts like a sieve, sifting the blood plasma to create the CSF. So it filters the blood to make CSF. It pulls out the water, it pulls out ions, and we have a whole choroid plexus video that you can check out for more details about this, including the tight junctions, the properties, and exactly how CSF is formed. So how is CSF analyzed? It's actually quite useful diagnostically, and it's extracted from a patient via lumbar puncture. And the reason is that it's a lot easier to access the CSF that surrounds the spinal cord than to access the CSF that's inside the brain, right? You don't want to puncture or poke the brain. So let's find the path of least resistance, and that happens to be at the very bottom of the spinal column, because in the human, the spinal cord ends at about L2, vertebral body L2. What's remaining here at vertebral body L3, L4, L5 is what's called the cauda equina. And that cauda equina is like noodles of nerves floating in this sac of cerebral spinal fluid. And so the clinician can go in. Typically it's done between L3 and L4 vertebral bodies, or between L4 and L5. But here it's being done between L3 and L4. The clinician goes in with a needle and can extract a small sample of cerebral spinal fluid. And so what do we do with that CSF? We'll do a CSF analysis. So normal CSF should have a certain opening pressure of about 7 to 18. The appearance should be clear and colorless, like water, shown here. So the healthy CSF should really just have water and ions and relatively low protein level. And this is the glucose level. It should be negative gram stain. There should be very limited white blood cells, etc. In the case of a bacterial infection, such as bacterial meningitis, there's going to be an increase in the opening pressure. One of the things that's most indicative here of the bacterial infection is this positive Gram stain, although that's in 60 to 90% of cases. But the appearance will be turbid. In viral meningitis, there'll be a negative Gram stain. The appearance tends to be clear, but there will be a difference in predominantly lymphocytes here, whereas in the bacterial there's predominantly neutrophils. As xanthochromatic is often seen in the case of a subarachnoid hemorrhage, SAH, where you're going to see an increase in red blood cells. You'll either see it'll be either grossly bloody or mildly, or even clear in some cases if it's a very mild bleed. Now this is something that you can just refer to as a reference. Main point is that it should be clear. So let's review here. Where do you find CSF? So outside the brain and the spinal cord here, so around the brain and around the spinal cord, all the way down. So we're not showing it here, but it is all the way down and around the spinal cord. We find this in the subarachnoid space. But what about inside the brain? Where do we find CSF? Inside what's called the ventricles, or the ventricular system. So let's talk about the ventricular system for a moment. So this develops from the hollow of the dorsal hollow neural tube. So when the brain develops, it starts out as a tube. There's these 5 secondary brain vesicles: telencephalon, diencephalon, mesencephalon, metencephalon, and myelencephalon. And inside each one of these, in the tube, the hollow of the tube ends up forming the ventricles. So this will form the 2 lateral ventricles. The diencephalon hole will form the 3rd ventricle. The mesencephalon hole will form the cerebral aqueduct. And then the met and myelencephalon will form the 4th ventricle. So here is what it looks like in the adult. So it's C-shaped, first of all, because that neural hollow tube bends several times in the process of development. And we end up with this ventricular system inside the brain. You can see the 2 C-shaped structures are called the lateral ventricles. In the midline, that's the 3rd ventricle. It's connected to the lateral ventricle here by a foramen, also known as the foramen of Monro. Then there is this cerebral aqueduct that dumps the CSF into the 4th ventricle, and then it continues on into the central canal inside the spinal cord, although that's only present. It oftentimes closes up. It's only present at about 30%. We have a video where you can check out the ventricles on MRI in the How to Read an MRI the Brain video. So here's another 3D view of the same thing where it's color-coded. So you can see the ventricles here in blue. So there's a right lateral ventricle and a left lateral ventricle. They're C-shaped, and each one of these projections is called a horn. There's an anterior horn which goes into the frontal lobe. That's called the frontal horn. There's the body. The posterior horn goes into the occipital lobe, so it's also called the occipital horn. And then the inferior horn down here goes into the temporal lobe, so it's also called the temporal horn. The foramen of Monro is shown in this teal color, the 3rd ventricle, which is sandwiched by the thalamus. And that hole inside the 3rd ventricle is filled by a part of the thalamus that connects the right and left thalamic lobes at the interthalamic adhesion. So remember that lateral ventricles are lateralized, all the other structures are midline. In yellow is the 3rd ventricle, in red is the cerebral aqueduct, like an aqueduct that's connecting water from the town to the village. And then in purple here is the 4th ventricle. And then there are 3 holes that connect the 4th ventricle to the subarachnoid space. The lateral holes, lateral is for Luschka, L and L, are seen here. Let me pause this for a second. We're looking at the posterior view. So here is the midline hole, Meninges, and the 2 lateralized holes, Luschka. And then the remaining CSF will travel down through the central canal of the spinal cord. It circulates by gravity, but also by pressure, and there is also pulsations of the CSF in the subarachnoid space that are managed by the pulsations of the blood in the arterial system. So here is the lateral ventricles, at least a piece of them. You can't really see the entire lateral ventricles. Why? Because lateral ventricles are lateralized, and this is a mid-sagittal cut. So you're cutting right down the middle. The left lateral ventricle would be going into the board. The right lateral ventricle would be coming out towards you. Here is the foramen of Monro that drains the CSF into this space for the 3rd ventricle. And then that CSF would drain into the cerebral aqueduct and into the 4th ventricle. So the body makes about 500 milliliters. That's like half an Nalgene bottle of CSF a day, right? Half a liter. That's a lot of CSF. And it's making it via the choroid plexus, which is in the ventricles, and it is producing it, but only about 150 mils of CSF can circulate at a given time. So how is this possible? Where does the other 350 mils go? Well, there is a constant turnover 2 or 3 times a day of the CSF. It's constantly lavaging the brain and washing throughout the system. So one of the things, common test question is to trace a drop of CSF from the lateral ventricles to the cerebral sinus. So let's do that right now. So here's the choroid plexus that's sifting the blood plasma to make this ultrafiltrate of blood plasma, CSF. We have choroid plexus in the third ventricle that wraps around, goes up the foramen Monroe into the lateral ventricles. So you have them in the lateral ventricle, you have choroid plexus in the third ventricle. You don't have choroid plexus in the cerebral aqueduct, and then you have choroid plexus in the fourth ventricle, right? So let's say that the choroid plexus, it can be made in any one of these places. It'll follow the same pathway of arrows. But let's say we have a drop that's made here in the lateral ventricle. It's going to go through the foramen Monroe right here into the third ventricle. It will then drain into the cerebral aqueduct into the fourth ventricle. If it's gone out of one of these foramens, it's going to come up and around into the subarachnoid space and lavage the brain. And then it will exit the subarachnoid space through this structure called number 8, arachnoid granulations, and dump into the venous sinus, also called dural sinus, also called cerebral sinus. All those terms are the same. What are these arachnoid granulations? Well, now we're looking at a coronal slice again here. This is the pia, this is arachnoid. The arachnoid makes this little pocket. It looks like cauliflower on a gross brain. And it allows for filtration and of the CSF to drain, because it's picked up all these byproducts of neuronal metabolism, including amyloid proteins and all sorts of other byproducts. And it's going to drain that and join back with the venous system. So inside the sinuses are venous blood. So here are veins that are also coming up and draining into the sinuses. And here is CSF that's draining through the arachnoid granulations into the sinus. Right? So the CSF is reabsorbed by the arachnoid granulations in the collection of venous blood between the 2 layers of dura mater, also called the venous sinuses. And all of these venous sinuses are named. The one that's mid-sagittal is called superior sagittal sinus. This is, again, another picture of the superior sagittal sinus. There's an inferior sagittal sinus. All of these sinuses are going to come together and drain into the internal jugular vein and take that venous blood and the CSF back to the heart. So there's a 3D resource here on Sketchfab. If you go to Sketchfab and you type in cerebral venous system, you can see this model. This shows all the different parts of the venous system as they sit in the skull. So number 1 is the superior sagittal sinus. 2 is the inferior sagittal sinus. 3 is the straight sinus. 4 is actually the great cerebral vein of Galen. We have the confluence of sinuses, transverse sinus. You can play around with this on your own and see how these are labeled. Okay, so let's just review our terms. We've got these ventricles. We've got the choroid plexus in the ventricles. We've got subarachnoid space. And then we've got sinuses, which are this space between the 2 layers of dura. So then what's a cistern? Because you'll hear this term cistern. Is this the same as a ventricle? No. Is this the same as a venous sinus? No. This is another space. And it's an enlargement of the subarachnoid space. So yes, it is the same as a subarachnoid space, it's just bigger. And because the pia, shown here in red, is so closely adhered with the brain, it goes into every possible groove. But the arachnoid, shown here in blue, doesn't go any, this is the bottom sheet in red, and this is the top sheet in blue. And so there's some places where there's going to be a space between the pia and the arachnoid. That's that subarachnoid space, which is going to be larger than normal. See, here it's a sort of normal size. Here it's going to be larger. And in those enlargements, that's called a cistern. There's the cerebellomedullaris cistern between the medulla and the cerebellum. There's the pontine cistern at the bottom of the pons. There's the interpeduncular cistern, that's in between the two peduncles of the midbrain. And these are the three major ones. And they're used as references primarily on looking at CT or MRI. Okay, so let's go back to our practice question, because we've answered a lot of questions. We've answered what's CSF, what's choroid plexus, we've talked about granulations, we've talked about CSF flow, we know now what the cerebral aqueduct is, and we know the meninges. So the only thing we need now to answer this question is this information about what's hydrocephalus and what's the difference between non-commuting and communicating. So hydrocephalus literally means hydro, water, cephalus, brain. So it means that there's water on the brain. So an enlargement, typically, of the ventricular system. So here is, and it often happens in children, although it does happen in adults for various different reasons. And you can see the ventricular system here in a neurotypical child. Here is a child with hydrocephalus. And you can see that there's an enlargement of the ventricles. There is flattened gyri because of the pressure and narrow sulci, and an enlargement of the head because these cranial plates have not yet fused, so the cranium can expand. And so in children with hydrocephalus, that when it goes untreated, you can see this grossly enlarged head. Here is hydrocephalus on a CT scan, so we're looking at an axial CT, and you can see enlargement of the ventricles here, and here is in a neurotypical brain. What is the difference now between communicating versus non-communicating? So communicating hydrocephalus occurs when the flow of cerebrospinal fluid is blocked after it exits the ventricles. The way to determine whether or not it's communicating versus non-communicating is to answer the question, does CSF communicate with the subarachnoid space? If yes, then it's communicating. In other words, the term communicating refers to the fact that CSF can still flow between the ventricles, which remain open. So this can be caused by an overproduction of the CSF. So CSF is being overproduced and it can flow into the subarachnoid space. That would be communicating. CSF, there's thickening of the arachnoid, and so it can't drain. That would be communicating, because the question is, can this ventricles still communicate with the subarachnoid space? Yes. Non-communicating hydrocephalus is also known as obstructive hydrocephalus. So if the CSF cannot communicate with the subarachnoid space, so you answer no to that question, then it's non-communicating. So this occurs when the flow of CSF is blocked along one or more of the narrow passages connecting the ventricles. The most common of these is stenosis of the aqueduct. So let's go back to our review question now. So in overproduction of CSF by the choroid plexus, can you communicate with subarachnoid space? You can. So that would be communicating. Obstruction of CSF flow by the arachnoid granulations, here, there's still CSF communicating between here and here. It's just blocked right here. So this one also would be communicating hydrocephalus. Stenosis of the cerebral aqueduct, that is the correct answer for non-communicating hydrocephalus. And inflammation of the meninges, well, if there's inflammation of meninges, it's going to irritate or cause problems with this point in the flow, but there'll still be flow between the fourth ventricle and the subarachnoid space. And so the correct answer here is C. Now, how do we fix this? Well, there's a couple different ways. One is to put in a shunt, pretty much like a little straw that's fed and through a tube, and that extra cerebrospinal fluid is drained into the peritoneal cavity. There's also a surgical procedure in the case of a stenosis of the aqueduct. There is third ventriculostomy, where a hole is punctured in the third ventricle, and that just drains directly into the subarachnoid space, so you bypass the aqueduct in the fourth ventricle.